Okay, welcome to this explainer. Today, we're diving into a fascinating systematic review that could literally revolutionize sustainable forest management. We are talking about completely shifting away from harmful chemicals and instead tapping into the microscopic ecosystems that actually help trees defend themselves. It's an absolutely wild concept, so let's get into it. I want to hook you right out of the gate with this massive number, 74. Specifically, we're talking about a 74% reduction in root rot disease severity. And here's the crazy part. That was achieved without a single drop of chemical fungicide. When you think about global forestry and the massive scale of disease, a 74% reduction is an absolute game changer. And this isn't just theoretical. It's rigorous, peer-reviewed science happening right now in labs and greenhouses. So here's how we're going to unpack that staggering statistic today. We'll quickly cover the conifer disease crisis, look at the co-inoculation solution, uncover the microscopic secrets of defense priming, explore the unique plant hormone network, and finally, look at what all this means for the future of forestry. Let's start with part 1, the conifer disease crisis. To really understand the stakes here, we have to look at the scale of the problem. Boreal coniferous forests cover around 1.37 billion hectares globally. They're absolutely crucial for carbon sequestration, biodiversity, and of course the timber industry. But they are under siege. Nasty soil-borne pathogens like Heterobasidion and Fusarium are just wreaking havoc. We're talking hundreds of millions of euros in losses annually. These pathogens rot the roots from the ground up, destroying everything from tiny nursery seedlings to established, mature timber stands. But here is the real kicker. Our standard chemical arsenal, it's just failing us. For decades, commercial forestry has relied heavily on broad-spectrum chemical fungicides, but pathogen populations are getting smarter, constantly developing resistance. Plus, strict biosafety regulations, especially over in the EU, are rightly restricting their use. And maybe the worst part is the collateral damage. These chemicals basically nuke the natural, beneficial microbial communities in the soil, the rhizosphere, that seedlings desperately need to stay healthy. So we desperately need a biological alternative, which perfectly tees up part 2, the co-inoculation solution. So for a long time, the go-to strategy was single-agent biocontrol, basically finding one good microbe to fight the bad ones. And hey, sometimes that works. But the data clearly shows that multi-agent consortia, meaning using a collaborative team of functionally compatible microbes, significantly outperforms those isolated treatments. It's kind of like the difference between sending in a lone wolf versus a highly coordinated tactical squad. When you combine microbes with complementary skills, the whole system becomes way more resilient against pathogen attacks. All right, remember that incredible 74% reduction in disease severity we talked about earlier? Here is the exact anatomy of how they pulled it off. Researchers took mass and pine seedlings that were actively being challenged by a deadly Fusarium pathogen. Then they brought in the squad. 2 specific agents. First, a fungus that secretes inhibitory organic acids, and second, a rhizobacterium that literally degrades the pathogen's cell walls. By combining these two, the plant gets direct pathogen suppression on the outside and an immune boost on the inside, all at the same time. That synergy is the magic behind the 74% success rate. And this isn't just some crazy one-off fluke. Check this out. In an entirely different system involving Norway spruce, researchers used a beneficial endophyte. That one addition slashed the root rot pathogen's active presence by an absolutely astounding 92%. The evidence for biological control is just undeniably potent when it's dialed in correctly. Let's shift gears to part 3, secrets of defense priming. So, how does this actually work on a cellular level? It all comes down to a strategy called defense priming. Think of it as putting the plant into a ready but restrained state. If a tree's immune system is constantly firing at 100%, it's wasting precious metabolic energy that it really needs for growing roots. Instead, beneficial microbes precondition the conifer so it acts like a coiled spring. It saves its energy, but the absolute second a pathogen attacks, boom, the plant launches a vastly faster and broader immune response than it ever could unprimed. And we actually have the rigorous transcriptomic evidence to back this up. When the beneficial microbes show up, they essentially tell the plant to start prepping its weapons. The plant upregulates its phenylpropanoid pathway to reinforce its cell walls, and it gets pathogenesis-related proteins like chitinase ready to literally chew through fungal invaders. It's heavily arming itself just beneath the surface. Now, researchers found that this microbial teamwork protects the plant in 2 very distinct ways.
First, there's transcriptional priming, that active, coiled spring immune boost we just talked about. But then there's ecological displacement. In this scenario, the good microbes simply get there first and crowd out the bad guys. Because the pathogen can't even get its foot in the door, the plant doesn't suffer any tissue damage. This leads to what researchers call transcriptional calm. The tree doesn't even have to activate its own genes because its microbial bodyguards handle the threat out front.
Moving on to part 4, the plant hormone network.
To really grasp how these trees communicate internally, we have to look at their hormone networks. And honestly, it completely rewrites textbook botany. A fascinating tale of two systems emerges when you compare standard flowering plants with the proposed model for conifers.
See, in your standard flowering plant, the two main defense hormones, salicylic acid and jasmonic acid, are antagonistic. If the plant turns one on, it actively suppresses the other. You can fight off one type of bug or the other, but rarely both at the same time. While conifers totally defy the standard. Their defense pathways are actually cooperative. They beautifully co-activate both systems simultaneously through a shared signaling hub. These ancient trees have evolved an incredibly robust way of maximizing their immune response.
Which brings us to our final section, Part 5, the future of forestry. Okay, keeping it real for a second. As amazing as a 74% or 92% drop in diseases, we do have to view this through a critical lens. A risk of bias assessment shows some gaps in how these experiments are currently reported. We still critically need better assessor blinding in the labs and, honestly, more operational field-based trials. The lab science is rock solid, for sure, but a wild forest is a much more complex, chaotic environment than a temperature-controlled greenhouse.
But despite those gaps, the roadmap for applying this out in the real world is crystal clear. If forest managers want to use these multi-agent microbial squads, they have to follow 3 rules. First, apply prophylactically. Timing is everything. The good microbes must be established before the pathogen ever shows up. Second, the microbes have to be functionally complementary, like our cell wall degrader paired with our acid producer. And third, you must verify that the specific tree and the specific pathogen respond well to the treatment. This is precision biology, not a generic spray.
So, I'll leave you with this to chew on. Given the devastating economic impacts of root rot, the massive limitations of standard chemicals, and the mind-blowing 74% disease reduction we can get just through symbiotic microbial teamwork, are we finally ready to replace failing chemicals with these highly tactical microbial consortia? The transcriptomic blueprints definitely say yes. Now we just have to take it from the greenhouse to the great outdoors. Thanks so much for joining me on this explainer, and keep questioning the science.